So with all that in mind, I'm going to very briefly introduce Helen. Professor Helen McShane is Validate's Network Director and the Director of Oxford NIHR Biomedical Research Centre. And this is at the beginning of a long list of other titles and accolades that, if I were to go through all of them, would waste all of our, this entire session. So this was the short introduction for Helen, and I'm going to hand over to her now, who's going to take over from this point. Thanks, Helen.
Blakely, super, thank you, and thanks so much for organising this, and thank you to everyone who's joined. I think this is an enormously exciting initiative, and we're very pleased to be able to do it. So let me just very briefly tell you a little bit about Validate. So Validate is a network of scientists, researchers, doctors, healthcare workers who work on vaccine development specifically for 4 different pathogens, 4 different bugs. So we work on tuberculosis, we work on leprosy, we work on something called Burkholderia pseudomellii, which is a bug that is particularly common in Southeast Asia and causes particularly lung disease. And we work on leishmaniasis, parasitic infection, particularly troublesome in South America and other areas of the world. And the reason why we picked these 4 pathogens or bugs is because they're all quite similar. They all live inside cells. And there are ways in which these bugs are all very clever, and they hide from the host immune response. So we thought it would make sense to put together people working on all those different pathogens to see if creating a network where people can talk to each other. We have meetings. Pre-COVID, we used to meet face to face and have presentations and scientific discussions, and now we do all this online, of course. And we hope that by bringing together people who work on all these different pathogens, we can make faster vaccines, the vaccine development against them. So that's just a little bit of introduction. One of the things we're very interested in doing in this network is to support the career development of the next generation of scientists who are going to lead these fields. And I'm delighted that my co-speaker, Rachel Tanner, Dr. Rachel Tanner, is a Validate Fellow. So she received a 2-year Validate Fellowship to support her becoming an independent scientist. So I'm going to start the presentation. So the format is I'm going to give you a short presentation. I'm going to talk just very briefly about my background so you understand what I've done in my career. I'm then going to talk a little bit about vaccines, and of course vaccines are in the news all the time at the moment because of COVID, and a little bit about tuberculosis. And then I'm going to hand over to Rachel, who's going to talk a little bit about herself, and then also she's going to talk a little bit about immunology. And then the floor is open for questions, and please don't be shy. We really would like to hear from you. So let me now share my screen. Blakely, can you just confirm you can see my screen?
Yeah, that seems to be working. Do put in the chat, everybody, if you can't see the slides, let me know.
Yeah, do. Okay, so I'm going to assume you can see that next slide. Is that all right? So this is entitled Serendipity, and I think there's a really important point here, which is that, you know, I didn't set out to end up the Professor of Vaccinology at the University of Oxford. And I think the point here is that it's difficult to know when you're at school where life will take you. And I think my advice for all of you would be to take the opportunities you're given and follow your nose, follow what you find interesting, because that's what I did. And I had a slightly complicated path. I remember being at your stage in the sort of, well, it was 5th year then, so 16, and I sort of thought, well, do I want to be a doctor or do I want to be a lawyer? And I wasn't really quite sure, but I then went into hospital when I was 16 to have an operation and realised that I really quite liked hospitals. They were quite fun places to be and be there, and actually I very much like people. I'm a very people person, so I thought, okay, I better be a doctor then. So I dropped the idea of being a lawyer. And I then applied to medical school, and I put there that was despite my maths teacher, who I went to a school in southeast London, which is in fact now closed. It wasn't a terribly good school, and my maths teacher basically told me that he thought I should probably be a housewife. And I don't think teachers say those kind of things to girls anymore, but I'm pretty stubborn, and that made me decide that I was going to go to medical school and show him. So I did. Really the rest is history. But the interesting thing is I actually started life as a GP. I went through all of my medical school assuming I was going to be a GP, a general practitioner, and did 6 months in general practice straight from my qualification. I quite liked it, but wasn't really quite for me. So that was the point at which I decided I would switch career. It's never, you know, there are very few things in life you do that are irrevocable. And if you start something and you try it and you realize it's not for you, then change. Life is long, and it's important to find something in life to do that really inspires you. So I then started doing some work in hospitals, and at the time I was on the south coast in Brighton, and this was at the beginning of the HIV epidemic in this country, and I had a ward full of patients who were all largely men, and they were all dying from HIV. This was before we had any treatment for HIV, and that was really what sparked my interest in infectious diseases. And it was a fascinating mix of people dying of rare, interesting infectious diseases because their immune system was very damaged by the HIV virus, but also palliative care. We were looking after people who were dying, and many of them were my age or younger. So that was fascinating, a very heady mix for a junior doctor. And of course, people with HIV are more likely to get TB. So that's the link with what I've now spent the rest of my research career working on, which is tuberculosis. So I work in the Jenner Institute in the University of Oxford. I've heard of Edward Jenner. Here's a picture of him vaccinating his gardener's son with his vaccine that he developed against smallpox. And of course, the only infectious disease we've ever eradicated is smallpox, and that was by effective vaccination. The power of vaccines, really. In the Jenner Institute, we have very much a global health agenda. So we focus on vaccines that make a difference. We focus on HIV, TB, malaria, dengue, pandemic influenza, and of course, as you all know, I suspect from reading the BBC website and the newspapers, we now focus on COVID as well. We focus on what we call translational research. So this is, we develop vaccines, but we're not just a scientific laboratory developing vaccines to test in a laboratory setting. We're actually trying to develop vaccines that save lives. So we're very good at moving those vaccines very quickly into clinical trials, testing those vaccines, testing them in Africa, testing them in areas of the world where the diseases we're interested in are most prevalent. And then ultimately potentially licensing those vaccines. We also have a One Health agenda. And what I mean by this is we're interested in vaccines for animals and humans. So, for example, tuberculosis is a disease that affects cattle as well as humans. And it is potentially the case that a vaccine that works for humans could also work for cattle. Vaccination is the most cost-effective health intervention. As I say, we've eradicated smallpox, the only infectious disease we've ever eradicated. We are tantalizingly close with polio, and I don't think the world will forgive us if we go back on that. And we have 27 vaccines licensed at the moment. I think actually now it's 28 now that the Pfizer-BioNTech SARS-CoV-2 vaccine is licensed. And I have HPV there as one of the most recent vaccines. And in the WHO, what we call the expanded programme on immunisation, up to 95% of babies throughout the world are vaccinated with a whole load of vaccines to prevent against many infectious diseases that without those vaccines, many babies and small children would die every year from those infections. It's estimated that 2.5 million lives are saved every year because of vaccination. So how do vaccines work? This is, I think, my only immunology slide, which I'm going to hand over to Rachel to talk about. Of course, they work via immunological memory. So what we do with a vaccine is we develop a vaccine that is either a weakened or killed version of the whole bug, or it takes just a few proteins from the bug. And some of you may have heard of the spike protein, which is the bit of SARS-CoV-2 people are including in the vaccines. And then we use that in a safe way to stimulate a primary immune response, which is quite slow and quite small. But what that means is that when you are then exposed, or someone who's been vaccinated is then exposed to that real pathogen, your immune system remembers that it's seen it before. So it then makes a response that is different in two ways. It makes a much faster response and it makes a much stronger response. The key things we look for in vaccines are obviously safety, because vaccines by and large are given to healthy people to prevent infection. We need to look at efficacy, which means, do they work? Do they actually stop people getting the bug that we're trying to prevent? And how long do they last? It's no good giving a vaccine if it only lasts 6 months. Ideally, we want vaccine-induced immunity to be lifelong. Broadly speaking, there are two arms of the immune system. So we have the humoral arm of the immune system that induces antibodies and generally works on pathogens that don't hide inside cells. And we have the cellular arm of the immune response, T lymphocytes, that work on pathogens that are very good, particularly at hiding inside cells. So we then need those T lymphocytes to kill those cells so that we can then kill the pathogen that's hiding inside them as well. And it's important to know which arm of the immune response you need to induce for different vaccines. So for tuberculosis, we know that the pathogen hides inside cells. And actually, although we think antibodies may play a little role, we definitely know that T cells are critical to protect against TB. So just a few pictures now to sort of get you spotting the abnormality. So here's a chest x-ray. This is an absolutely typical chest x-ray of someone with TB. So in the middle, I don't know if you can see my pointer, you have the heart, this sort of slightly irregular shaped thing in the middle. And then you can see on the right of your screen, you have the lung field, which is essentially black. It's black because there's air in it, and that's good. You need air to be in your lungs. But you can see that there isn't much air here in this upper, on your screen, left side. And that's because there's a great ball of pus there, and that's a tuberculosis infection. TB always infects, or typically infects, the upper parts of the lungs because it is what we call an aerophilic organism. It likes oxygen, and there's more oxygen in the top parts of the lung. But you can see that in this picture, although that is clearly very damaged, the rest of the lungs look normal. In contrast, this picture, you can see that both the lung fields look pretty abnormal. This is called disseminated TB, when TB has spread throughout the entire lung field. This person is pretty sick. And then finally, and this is a chest x-ray I show the medical students to tease them, in this abnormality you can see some very, very regular circles in the top of this lung. And people sometimes think they're cavities that have formed because of TB, but actually this is a form of treatment. This is essentially inserting polystyrene balls between the two layers of pleura that surround your lung. And what that does is it squashes that bit of the lung. If you squash that bit of the lung, no oxygen goes in, which means the TB inside that bit of the lung dies. And this is a much easier way to treat TB than removing the whole lung. And you might think that's just historical and something we used to do before antibiotics, but actually now that we have multi-drug resistant strains of TB, which sometimes are impossible to treat, people in areas of the world, particularly in Russia and Eastern Europe, where we have TB that is not treatable anymore, are going back to surgical methods of treating TB. So TB is sometimes called consumption in the literature. This is how it used to be described. And before we had antibiotics, people were sent to sanatoria in the Swiss mountains where they'd sit on the balcony and get sunshine and probably some vitamin D from the sunshine that would perhaps help some of them, their own immune system, control the TB. Of course, some people died and not everyone could control TB without antibiotics. So TB, it's really important to remember there's a lot of focus on COVID at the moment, and we know that about 1.5 million people have died of COVID now, which is terrible. But actually 1.5 million people die of TB every single year. And this is a disease that's not been around for less than a year. It's been around since the pharaohs. So and the part of the reason for that is TB is a disease that is linked to poverty. We know that the poorer you are, the greater your risk of TB, and essentially TB rarely infects presidents and prime ministers, and there's been really decades of little funding going into TB research, which is why it remains such a very, very big problem today. TB drugs, so we treat TB with 3 different drugs. You can see that the early drugs were developed really quite a long time ago now, but the important thing about this slide is to see the gap. Between 1957, rifampicin, which is one of the most important drugs we use today, and then 2012, before we had any new drugs licensed. A huge gap where we frankly thought TB was over, but it clearly isn't. We use very old diagnostic tests. We inject some proteins from TB into the skin to see if you get an immune reaction in your skin. That's one of the ways we diagnose TB. And we look down the microscope to look at the sputum, and you can see here these red, what we call acid-fast bacilli, and that's absolutely characteristic of TB. This person is quite infectious. You wouldn't want to sit next to them on the bus. And this slide shows you how many vaccines were in development being tested in people in the year 2000, 20 years ago. And it's deliberately a blank slide because there were none. And that is really one of the reasons why TB is such a big problem today, because there was such a lack of investment in research and development for tools to control this pandemic. We now know that there are lots of vaccines. There are about 14 or 15 that are now being tested in clinical trials, and some of them are listed here, and that's fantastic. But it's really important we keep this going, we keep this momentum going, if we are to tackle the TB epidemic anywhere near as effectively as we have tackled the SARS-CoV-2 pandemic. So finally, just to finish, I've done lots of clinical trials in my time in this role. We've done a lot of work in South Africa, down here at the bottom of the map. We've done some trials in Senegal and the Gambia, and we're currently doing a study in Uganda. And I've had the absolute privilege of working with fantastic doctors and nurses and scientists at all of these sites to build up fantastic academic collaborations, really working together to try and develop a vaccine for this terrible pathogen that really, really does need new tools to help us control it. So that was just a brief illustration of what I've spent the last 25 years doing. I'm now going to stop sharing my slides if I can. And then and now I'm going to hand over to Rachel, who's going to tell us a bit about herself and about immunology before we open up for questions. Rachel.
Thanks. I'll just share my slides, hopefully. Okay, can I just check that with Blakely that you can see that?
Yeah, can see it.
I can see it. Everyone else, let me know if you can't see it.
Great, thanks. Okay, so my career is less exciting. My career path is less exciting and illustrious than Helen's, obviously. I'm not a medic, so I started off doing biology as my degree. And I think I decided at school at some point that I wanted to be a scientist to do with medicine. Because I wanted to help people, but I'm a bit of an introvert, so I think I would prefer to be on my own in the corner of a lab pipetting than having to deal with people all day, every day. So I kind of had this vague idea, but I didn't really know what being a scientist entailed, as I think nobody really does until you get a chance to go and see it and experience it. So one of the things I would really recommend if you're thinking about going into science is to see if you can get a brief work experience or a shadowing or something like that. I think previously these were only really open to people who knew someone who had a parent in the field or something. But now there's lots of schemes and different ways that you can go into a lab. So I would recommend just Googling, find something in your area and just go and have a look and see if it's something that would be for you. Luckily, it turned out it was something for me. And I'll talk a bit more at the end about why I like being a scientist. Okay, so to give you an overview of what I'm going to talk about, I'm going to start with some pretty hardcore T-cell immunology, so right at the deep end, and then look at how we can apply that to design a vaccine. So by the end of this, you're going to be expert vaccinologists. I'm going to talk a bit about sweaty T-shirts, and when I get to that point, I'll explain why. I'm also going to talk about how we test TB vaccines and the 3 Rs, which is about trying to reduce the use of animals in vaccine testing. And then, as I mentioned, why I like being a scientist, and I'll show you some holiday snaps, which I'm sure is what everybody came for. So TB is these little rod-shaped bacteria, so they're represented here in green. Obviously, in real life, they don't have eyes, but their cell of choice to infect is a macrophage, which is a type of white blood cell. So if you come across somebody who is infected with TB and they cough in your vicinity, you might inhale some of these TB bacteria and they'll go into the lung, and then these macrophages, which are represented by this purple cell here, will engulf those cells, engulf those bacteria, and then the bacteria will happily live inside that macrophage and replicate and cause an infection. Okay, so you might have noticed that this macrophage has these little blue things on the surface, and these are called MHC. So when the macrophage gets infected, it chops up the bacteria into lots of little pieces. So Helen mentioned earlier about proteins. Some of these pieces are proteins, and what the macrophage then does is it presents these proteins on the surface bound to the MHC. And that's like a cry for help to the rest of the immune system. So it's telling the immune system that it's infected. When it does that, along comes a T cell, and T cells have these receptors on the surface shown in red, and they look out for these bits that are presented by the MHC on the surface of cells. So they're looking out for them, and if they find one that they recognize, then they can bind to it. What happens when they bind to it? Well, they make lots of copies of themselves, so you end up with lots of T cells, and then they can do different jobs. One of the jobs they do is to destroy the infected cell. So they'll break it up, the cell will die, but so will all the bacteria inside it, and that helps to clear the infection. But not all the bits that the macrophage puts on the surface can be recognized by the T cells. Different T cells will recognize different shaped bits. And so, for example, here the macrophage has chopped it up into these different shapes. One of them is this triangular bit. The T cell, its receptors don't recognize that, they can't bind to it, and it doesn't know what it is, and it's not able to respond. So how does this relate to vaccines? Well, as Helen mentioned, we can design vaccines that have these bits inside them. So the bits that or the proteins or whatever other bits that the macrophage has chopped up and put on the surface, we can use those in the vaccine. And if you remember this triangular bit, which didn't induce a good T cell response, if we use that in our vaccine, it wouldn't be very useful. So you'll just end up with no effective T cell response. If you use a different bit, so the bit that we saw originally that the T cell could recognize, then it will recognize it, it will produce lots of copies of itself, and those T cells will hang around in your body for a really long time because they produce something called memory, and they'll be there ready to recognize any real TB if you come across it during your life. So that's what we're trying to do when we're making new vaccines against TB, is to identify which of these types of bits would be most useful to put in the vaccine to induce a good T cell response that will make lots of copies of itself and remain for as long as possible within the person that's vaccinated. Okay, so get a little bit more complicated now. You get different types of MHC on the surface of the cell, and not all MHC can present all the bits. So some MHC will present some, and some MHC will present others. If you have MHC that are unable to present anything, then obviously the T cell can't respond. It doesn't know that the cell's infected. It thinks it just looks fine. So what does this have to do with sweaty T-shirts? Well, the more different MHC you have, the more different bits you can present of different pathogens. So if you got infected by different viruses, different bacterias, the cells would chop them up into the bits and they would put them attached to the MHC. And if you have lots of different MHCs, then you can present lots of different bits of lots of different pathogens. And that means that you can have good T cell responses to all of those bugs and clear them from the body. And the MHC that each person has is a mixture of what their parents have. So if you have two parents that have the same types of MHC, then the offspring will have all the same as well. If you have a parent with one type and another parent with another, then the offspring will have a mixture of the two parent types. And remember that the more different types the offspring has, the more different bugs they can clear. So in 1995, there was a scientist in Switzerland who did an experiment where he got 50 men to wear a T-shirt for a couple of nights, so they got all sweaty and smelly, and then he sealed those T-shirts into containers, and he got 50 women to come along and smell the T-shirts and rank how attractive they found them. And they didn't know anything else about the men, so they didn't have any other information, what they looked like or anything else. They just ranked whether they found the smell attractive. And what they noticed is that different women had completely different ideas about what they thought was attractive. So some ranked a particular T-shirt as very high, the most attractive smell that they came across, whereas another would rank the same one at the bottom. And this turned out to be related to the MHC genes that those. So if the women had very different MHC genes from the man who was wearing a T-shirt, they would find the smell attractive. Whereas if they had the same MHC genes, they found it unattractive. And lots of different scientists have repeated this experiment over the years in different populations. And usually, not always, but usually they find a similar relationship. And so obviously there's lots of other things that play into human attraction and who we fancy. But this is probably one part of it, one component is to do with immunology. So immunology isn't just about saving you from diseases and from dying, it also has a role in who you fancy as well. Okay, I'm going to tell you briefly a bit about what I did for my PhD. So my PhD was really all about the 3 R's, which is trying to either replace, reduce or refine the use of animals in medical research. So we all know that animals are a necessity in medical research. You can't put a new drug or a new vaccine straight into a person without checking that it's safe and that it works in an animal. And so it has to be done, but if it has to be done, then we should try and do it in the best way possible. So we should try and use the least number of animals that we need and reduce the amount of stress or pain that those animals go through as part of the experiment. So when TB vaccines are tested, we usually start by testing them in mice. And so you might have a group of mice to begin with, usually around 6. They each receive the vaccine, and then once they've been vaccinated, they get infected with the TB to check whether the vaccine has protected them from getting the disease. The idea with my PhD work is to try and find another way of checking whether the vaccine's working so that the mice don't need to be infected. So here they get the vaccine and then blood samples are taken, and it's the blood sample in a test tube that's infected with the TB in the lab rather than the mice themselves. So the mice don't get sick, they don't have an infection. And TB varies around the world, so different parts of the world have different strains of TB. So the type of TB that you find in South Africa might be different from the one in India and different from the one in Russia. And so if you want to check that the vaccine is working against all these different strains, normally you would need to vaccinate a group of mice and infect them with one strain. You then need another group to vaccinate and infect with another strain, and then another group to vaccinate and infect with the third strain. However, if you use the test that we've been developing, you can vaccinate your one group, take some blood, and then test all of these strains in multiple different experiments in the lab without having to use any more mice or to infect any of the mice with any of the TB strains. So if you're interested in finding out more about the 3 R's, This is an article that I wrote in a journal called Frontiers for Young Minds, which I would recommend for all other science, as well as reading about the 3 Rs. And you can find it by searching in Google. And this is a scientific journal, but it's written specifically for young people. And so scientists try to relate their research in a way that's understandable. And it's also reviewed by young people, so they give us feedback on how to alter the article. Okay, so I'm just going to finish with why I think being a scientist is cool. I think probably the main reason is that if you're curious about things, instead of having to go and look them up, you can design an experiment and find the answer yourself, hopefully. And the answer isn't always what you're expecting. So sometimes you get an unexpected result and that takes you down a whole nother route of investigation. I also, as I mentioned before, I wanted to do something constructive to help people. Medical science is a great way to do that. You can have a huge impact. The types of diseases that we're trying to develop vaccines for can really save millions of people. Also, it's pretty flexible, so it's not like an office job where you kind of sit at a desk all day, every day from 9 to 5. You do lots of very different things. So one day you might be in the lab doing an experiment. The next day you might be writing a paper or a grant or helping a student to design experiments or lecturing undergraduates or going to conferences. So it's really varied. It's flexible in terms of the time. So sometimes we keep really bizarre hours, but that can also work in your favor. So it can work around other things. It's also collaborative, so you work with lots of different people, especially different groups all over the world. And related to that, I just wanted to note something that was unexpected to me really when I went into science. I didn't realise that science also gives you the opportunity to travel loads, so to conferences and meetings, which are often held in really interesting, cool places. Also to go and work with people in other labs around the world. And as Helen mentioned, doing field work or trials in other countries also it's given me the opportunity to go to lots of places that I otherwise probably never would have had the chance to. Cool, I think that's all I have for now.
Fabulous. Thanks, Rachel. That was lovely. So I hope you... Oh, sorry, there's a bit of a funny echo. I hope that's going to go. I hope you all heard that and saw the slides. Okay, I could see in the chat, I think there was one group that couldn't see the slides, but Blakely has sent them over. So I think we'd like to open it up for any questions. Rachel and I are here for the next 25 minutes or so, and we'll be really happy to take any questions. And please don't feel shy. You know, they won't be silly questions. And actually, part of the trick is to learn to ask questions, because you know, it shows that you're engaged.
Yes, so everybody, feel free to, if you can, and you've signed the consent form and everything, turn on your webcam so we can see you. We don't need to hide you anymore, but if you could stay on mute unless asking a question. Anybody have any questions, you can either type it in the chat or raise your hand. Do we have any questions from any of the groups or any of the individuals at home? This is always the most awkward one, is the first question. So who's going to be the first through the breach?
Go on, do be brave. How long does it take to design a vaccine? Well, that's a really important question. Oh, here they come pinging in. So the answer is, how long does it take a vaccine? Is sort of the answer to that is, how long have you got? And there are some vaccines which are really difficult and slow, like tuberculosis, and we've been working on that for a very long time. But there are some vaccines that happily, and of course you'll all know this now, turn out to be relatively easy. And what I mean by that is that the immune response that is needed to be induced is particularly the antibody response, the humoral immune response. So normally it takes 10, 15, maybe 20 years to develop a vaccine from start to finish. With our SARS-CoV-2 vaccine, and we now have 3 of them that have shown that they are efficacious, so they work. Clearly that's taken about months. And that isn't because corners have been cut. The SARS-CoV-2 vaccine has been done so quickly, and I say SARS-CoV-2 because that's the virus. COVID-19 is the disease, but it's the same thing. And the reason why it was so fast is not because any corners have been cut at all. Every step has been followed, but wherever possible, things have been done in parallel, so in the same time. So the animal studies that Rachel talked about have been done at the same time as the human studies, rather than normally what happens is the animal studies come first, and then only if a vaccine looks good in those animal studies do you then go on to do studies in humans. So doing things in parallel speeds things up. It's also the case that everyone who's been working on the SARS-CoV-2 vaccine programs has been working 24/7, including the regulator.
I think we might have lost Helen. Rachel, do you want to take? Oh, Helen, you back?
No. Oh, sorry. Yeah, can you hear me?
Yeah, you're back. Rachel, do you want to?
Yeah, do you want, I'll take another question, I think.
Okay.
Oh, I can. Yeah, I can. Oh no, Helen's back.
Helen's back.
I'll move. Can you hear me?
Yes, we can hear you now.
Sorry, I'll move. Just normally it takes.
Right, Helen, if you.
Sorry, I'll carry on.
If you stand down a second, and Rachel will take over. My student, there's lots of questions coming in. My students want to know. Oh no, that was someone else. Why are people against vaccines? Is one of the questions there. If you don't mind taking a controversial one there, Rachel.
Yeah, I think a lot of the reason people are against them is because they don't understand aspects of it. So maybe they've read something, some misinformation on the internet, or there's just something that they're kind of skeptical about because it doesn't make sense to them. So I think educating people so that they understand and they can make an informed decision is really important. Ultimately, it's free will, so vaccines aren't compulsory. It's up to individual people whether they decide to get them. But I think in order to make that decision, they need to have all the information, and it needs to be the correct information. So not something that some random person who doesn't understand it has posted on the internet, but the actual true information so that they can make that judgment.
Here's another question, and I've always wondered this. I can explain. I'm not a researcher, so I'm probably at the same level as everyone else in the seminar. How does a T cell memorize a pathogen, or how does the immune system generally do that? I think would be my question. So how does T cell memorize a pathogen? How does that work?
I might leave that to Helen.
So what happens is when you vaccinate someone, you induce a T cell response. And what happens is those T cells expand very quickly. That's what we call an effector response, because it's ready to do something. But of course, after a vaccination, it doesn't need to do anything. And then what happens gradually over time is the T cells contract and they convert to what we call memory T cells, so that they are, sorry about the dog, they are programmed to stay around actually potentially forever. But one of the questions, as I said in my talk, is how long do vaccines work? How long does that memory last for? And ideally we want vaccines that last forever.
Okay, now does anybody want to ask a question audibly? Maybe Rian, is there anybody in your is there anybody who would like to sort of ask a question using the mic? Click raise your hands or just say so in the chat.
Oh, hi Blakely. Can you hear me?
Go ahead, Rian. Go ahead.
Yeah. Nathaniel, one of the boys, has asked, what methods are used for distributing vaccines in less developed countries?
That's a really important and very topical question, and it's really important, and you will see that this is something that Oxford were committed to, or are committed to. COVID-2 vaccine is supplying the developing world, low and middle income settings. And what we there are a number of mechanisms by which we do this. WHO, there's a thing called the Global Fund, which funds distribution of vaccines around the world. UNICEF. So there are a number of non-governmental organisations that governments around the world feed into and pay to undertake to deliver vaccines around the world. And it's clearly really important that that happens. I think what's one of the things that is most obvious now with the COVID pandemic is that we live on a small planet, and if you have an outbreak of a new pathogen in China, it can be around the world in a week, in a few days. And as I often say to people with TB, you can be in KwaZulu-Natal in South Africa and have multi-drug resistant TB and be in Heathrow 12 hours later. So we live on a small planet, and it's really important that we have a commitment to give vaccines around the world, particularly to the poorest of the world's poor.
Thank you very much, Helen.
This is entirely up to you, Rachel and Helen, as to how much detail you want to give in this answer. But Anna Law has asked, how much money does a medical student, a medical scientist earn?
I think it varies is the answer, depending on what level you're at. So obviously, as you're more senior, you earn more, and as junior, you earn less. What is, I mean, so I think a junior scientist, probably earns 30k to start with, 25 to 30k.
That's a good enough answer. Your dog is lovely, from Sam.
Yes, I'm sorry about the dog. I'm moving around the house to manage the Wi-Fi, but
no, I think we're all happy with the dog, so no apologies needed. Here's a broader question, and I guess this is one of those piece of string ones, but this is from Eddie. What is the general financial budget needed to develop a vaccine?
That's it. The answer to that depends on how difficult it is, but the real cost is in doing those large phase 3 trials to show that, and it's usually tens of millions, if not hundreds of millions, to actually get a licensed vaccine.
And here's from Beverly. Is a vaccine guaranteed, especially for elderly of multimorbidity? Isn't it best to boost their immune system rather than vaccinate? So yeah,
so the answer, the question is, does a vaccine work in the elderly? And the answer is some do and some don't. We do need to make sure that our vaccines work in the elderly, particularly as they are the most vulnerable population for SARS-CoV-2. You can do that by looking at the immune response that vaccines induce. We know that people's immune response does decline with age, and the elderly do have a less effective immune system than younger people. There's not an easy way to boost that, but a good example is, so for example, in people with HIV, we tend not to give vaccines to people with HIV if they are very immunosuppressed, because they won't work. We tend to wait until we've given them antiretroviral therapy to boost their immune system before we vaccinate them.
Mr. Brown, you have your hand up. Do you have a question from your group?
Yes, this is a student actually. But are you guys worried that the virus will mutate like we've seen in minks before all vaccines are given?
Rachel, do you want to take that, or do you want me to take that?
I can try, and then you can add if yeah. What they found with SARS-CoV-2 is that the mutation rate is relatively slow, and so compared with something like flu, which mutates every year, the SARS-CoV-2 virus is slower, and so I don't think there's too much concern. And the other thing about the way you design vaccines is that you design them to target an antigen that it's very difficult for the virus to change, and so different parts of viruses can mutate, and some mutate without an issue for the virus, but other parts, if the virus mutates it can no longer survive or replicate, and so there's too much of a cost to the virus to mutate it. And so if we target the vaccine at one of those types of antigens, then it's very unlikely to mutate over time and make the vaccine no longer work. I
have a really excellent question here from Moreton Hall. which is a school, not a person. Do you think the way the COVID vaccine has been made so fast will speed up the way vaccines are made in the future or revolutionize it?
Yeah, that's a really important question. I mean, the answer is obviously we hope so. I think we have learned a lot from the way things have been done here, and things can be done in parallel, as I explained, rather than taking sequentially. But you have to understand that that's then done at risk. The reason why things are usually done sequentially is if things don't work in animal models, they don't progress to human trials. So if you do everything all at once, yes, it's quicker, but it costs more and the risks are higher. So I think there is that caveat. I think the other caveat is we've learned faster ways of working, but I'm not sure any of us want to continue spending the rest of our lives at the pace we've all been working at over the last 8 months. And I don't think regulators are going to start working 24/7 for the rest of time either. So you can speed things up, and I hope that we've learned a lot from this that will speed up vaccine development. But I don't think it's sustainable to work quite at this pace forever. Why is COVID-19 so slow to mutate? Rachel, I'll give you that one. I don't know that we know, actually.
Yeah, I'm not sure that's known, but there's like a variation between different pathogens in their mutation rate. So, for example, something like HIV mutates really, really quickly, even in a single patient. And so that's one of the reasons it's been so hard to make a HIV vaccine. But I guess it's fortunate that SARS-CoV-2 isn't like that.
Yeah, exactly. It's a very good question, though. I mean, you know, it's very difficult doing all this with a virus that we know so little about. It just hasn't been around for very long. So there's a question here. What was the difference? What was the difference in synthesizing the newly found COVID vaccine compared to previous vaccines? Is the basis still around mutation rate? So there are two ways to make a vaccine. You either take the whole bug, the whole virus in this case, and you kill it or you weaken it and use that as a vaccine, or you take, as Rachel says, some strong proteins, and all the subunit vaccines, which is what that is called when you take just one or a few proteins, have taken what we call the spike protein from COVID, and that does seem to be very conserved, so it doesn't seem to mutate. Now that's the way you make vaccines in general. One of the new ways to make a vaccine using RNA is a relatively new way, and in fact the Pfizer-BioNTech RNA SARS-CoV-2 vaccine is the first licensed RNA vaccine, and I think there are going to be a lot of people looking at whether RNA as a way of delivering naked RNA to the body that then encodes for that protein is a good way to make vaccines against other pathogens as well.
We have two TB-related questions, and everybody do put your hands up if you want to ask a question with the mic. Two linked ones, I guess, is why is the existing BCG vaccine so ineffective? And following on, that's from Kirsty Holland, and Rian has asked, why did TB vaccination programs stop in schools during the 90s?
Okay, I'll take the schools one, and then Rachel, just to share them out, can do the why is existing BCG so ineffective. So the TB vaccination program stopped in schools in the 1990s because there was very little TB in this country. In fact, what happened was it didn't stop completely, but the program to vaccinate 13-year-old school children stopped. And in fact, what has continued is that at-risk babies are given BCG. So it isn't that there's no BCG vaccination in this country, it's just that it's only given to people who either live in a city where there's lots of TB, so babies in London get BCG, babies in Manchester get BCG, babies in Birmingham get BCG, because there's a lot of TB in all those cities. But the adolescent school program, which cost a lot more to deliver, was stopped. And in fact, the best evidence that BCG works is that it's given in babies. So in fact, what we have now is a better program in that it's given at the age that is best to show protection.
Sorry, there's lots of questions coming in. And
Rachel, go for the one about why BCG doesn't work.
Yeah, so BCG works differently in different parts of the world. So, for example, in the UK, it's quite effective, around 80%. But in other parts of the world, so particularly around sub-Saharan Africa and India, for example, it's really, really low. It seems like it doesn't really confer any protection at all against TB. And we don't know for sure the answer, but I think the leading hypothesis is to do with exposure to other types of bacteria that are very similar to TB. So there's something that we call non-tuberculous mycobacteria, and they live in the environment. And the closer you get to the equator, the more of those types of bacteria there are in the environment. And so if people are exposed to them growing up, then they develop an immune response to those bacteria, and that seems to block the action of the BCG vaccine. And so unfortunately, because a lot of the countries that have a high rate of TB are in those areas around the equator, it means that BCG is the most ineffective in the areas that need it the most. And so that's why we really need a new TB vaccine.
Blakely, I'll take this one. Is it true that the BCG provides some protection against COVID? The bottom line is we don't know. For a long time, there's been an idea that BCG might work against other infections, particularly in low and middle income settings. And there's some data from West Africa to suggest that BCG reduces what we call all-cause mortality in babies. That means overall deaths, not just due to TB. This is a very controversial area. Not everyone agrees with it, and there is some data to suggest that that is not necessarily the case. So we don't know if that's real or not. Some studies show that it is, some studies show that it isn't. We know that you can give BCG to treat bladder cancer. So people inject BCG into the bladder, and we know that it is effective at treating bladder cancer. So there is some evidence for this. So the answer is, how would you test to see whether BCG protects against COVID? Well, you do a clinical trial. So there are 2 randomized controlled trials where people are being vaccinated with BCG or not, and then being followed up for a long time to see what their risks are of getting COVID. Those kind of proper, really carefully controlled clinical trials that we can actually properly answer that question. So the answer is we don't know yet, but we will know at the end of those trials.
So I can take the one on CRISPR. So somebody's asked, could CRISPR ever be used to protect against viral infections instead of traditional vaccine route? And this is an interesting one because in 2018 there were some scientists in China who attempted to do exactly that. So there's a particular receptor on the surface of cells that HIV uses to enter cells and infect them, and they use CRISPR to edit those receptors in twin girl embryos, and so those girls are gene edited to be resistant to HIV infection, but that raises a lot of ethical issues, and it was quite a scandal at the time, and obviously it needs to be very carefully regulated because it poses lots of questions around where you draw the line in terms of what can be edited and what can't be, and also the off-target effects. So people have suggested since then that actually people who have that gene edited, although they might be resistant to HIV, they have a shorter lifespan in general and are more susceptible to other types of infection. And so we really need to carefully understand the effects of CRISPR in a wider sense before that can really be pursued anymore. Yeah,
exactly. Shall I take this one?
Yeah,
one
more,
Blake.
and I think we need to start wrapping up because it's rapidly approaching 10 o'clock. So
let me take this one about animals. Are animals used to test drugs and vaccines due to similar genetic to human or completely different? So it's a very good question. So we testing things in mice usually, or small animals, and then depending on which pathogen you're using, you use different animal models. So for example, in SARS-CoV-2, hamsters are a very good model, and non-human primates are a good model. In TB, we use guinea pigs and non-human primates. We also use cattle. So there are different animal models depending on which pathogen you're working on. And in general, all of those animal models are used because they recapitulate, or they're the same as some aspect of human immunity, but none of them are usually perfect. So they're very important because it's really important to test the safety of something in animals before you start injecting it in humans. But one of the things we're quite interested in doing in my group, as are a number of groups around the world, is developing what we call human challenge models, where we develop very safe ways of giving humans infections excuse me, to test vaccines, because of course then you're testing your vaccine in what we call the target species, the people that or the species that you actually want to give your vaccine to. And there's a very well established human challenge model for malaria, where people who develop malaria vaccines can vaccinate human subjects, deliberately give them malaria, put mosquitoes that are infected on their arm in a coffee cup, get 5 bites, and then you follow them up very, very carefully to see who gets malaria. So those models are really useful, excuse me, and actually complement some of the animal studies we do. But everyone who does animal research in this area obviously follows very, very strict government guidelines. The UK guidelines to do animal research are the strictest in the world. But it is an essential component of developing safe medicines and vaccines for humans.
Yeah, thank you very much, Helen. We've only got a couple of minutes, so there's a we obviously can't do it audibly, but I'd very much like to thank Rachel and Helen for those excellent talks and some fantastic answers. I'd also like to thank everybody else who attended and asked some, I think Helen and Rachel would agree, some really, really good questions there. It was really, really interesting. What I'm going to do is I'm going to cut and paste all of these questions, and I'm going to try and get some answers, and I'll pop them in an email to everybody as follow-up. Just some quick things. Validate has 400 researchers in 59 countries, and I'd really like to do another one of these, Validate for schools. It doesn't have to be with somebody in the UK. We have loads of people in lower and middle income countries. So do get in touch, validate@ndm.ox.ac.uk. So drop me an email if you would like to arrange another one. That's for teachers, and maybe students might want to get a few of you together, etc. And we can put you in touch with some really, really interesting people. But other than that, with 1 minute to go, I'd like to say thank you one more time, and I look forward to being in touch with everyone in the future.